Welcome, ladies and gentlemen. Today I would like to talk about imine reductases, or shortly IREDs. I read about more than 30 papers, and here I will summarize all the important details about this enzyme class. IREDs can catalyze the imine reduction reaction, therefore they use iminium ions as a starting material to reduce them to the chiral amine products. Therefore, they also use NADPH as a cofactor. The iminium ions are rather unstable in nature because the carbon-nitrogen bonds are rather prone towards the hydrolysis, and therefore only cyclic imines or ones with carboxylic residues are natural occurring. Here, the iminium ions can be stabilized by the intramolecular hydrogen bonds by this carboxylic acid group, for example. So where can we find these fantastic amine products of the imine reductases? We can find them now nearly everywhere. We have it in very pharmaceutical relevant drugs, for example, codeine, which is deriving from the plant Papaver somniferum. Then we can have it in mefloquinine as an antimalarial drug, or in rivastigmine as anti-Alzheimer drug. The first imine reductases were discovered by a Japanese group where Mitsukura found in Streptomyces sp an R-selective imine reductase and an S-selective imine reductase to catalyze the 2-methyl-1-pyrroline to either the R enantiomer with a rather good conversion in enantiomeric excess, as well as to the S enantiomer, depending on which IREDs they used. Based on the research of Mitsukura, Nick Turner and Gideon Grogan and many more investigated different kinds of substrate scope for the imine reductases. Therefore, here they published a very nice review about 5-membered cyclic imine, 6-membered and 7-membered ring structure, as well as aromatic imines, which are accepted by these two R and S selective imine reductases. Our group also, Jörg and Stefan, characterized these two enzymes, and they figured out with different kinds of imine substrates they could also get rather nice conversion as well as an enantiomeric excess. In addition to these two imine reductases, different groups found also other imine reductases, which is reviewed in this paper here. For example, here's an S-selective imine reductases from Pseudomonas lactis. Then we have one in Halephilia bacteria and in many other bacterial strains. And they can also accept a bunch of different substrates, leading to very nice product yield as well as enantiomeric excess. In 2014, Huber et al. Found out that imine reductases are not only limited on the imine reduction, but they can also catalyze the reductive amination. Therefore, they use this carbonyl substrate and an amine donor, which was used under large excess, so there's the limitation of this amino donor, to yield the imine intermediate, and then they used the S-selective IREDs from the Japanese group to get the final amine product, where they have a decent enantiomeric excess but a very low conversion. Building up on this research, different groups were also trying to combine different kinds of carbonyl substrates with lower excess of the amine donor, For example, here they used a lower amount of methylamine, as well as here another group used ammoniac with a different ketone. And they also had different kinds of amines used during this for the reductive amination. For example, here propagylamine was used, and also a recent study, an imine reductase, where they used cyclohexanone and lulamine were under equimolar ratios to yield these products in rather good yield and enantiomeric excess. Imine reductases are NADPH dependent, and the cofactor is oxidized to NADP+. Different research groups also investigated to recycle this cofactor. And here Nick Turner investigated in different kinds of enzymes, and they recycled it with a glucose dehydrogenase. Therefore, in this paper, they investigated in different carbonyl compounds, and they used methylamine to yield to the chiral amine products. There they have a rather nice conversion with this product here. There they also used secondary amines and also aromatic substrates, and also protected amines they could obtain. Another paper was published from Montgomery in 2020, where they used also the glucose dehydrogenase and different kinds of combinations between the carbonyl compounds and the amines to yield the chiral amine products. Here they used higher concentration of amine donor and still got rather nice conversions with different kinds of imine reductases that they tested. And yeah, they have also different kinds of amine products that they could obtain during this screening. So how can this imine reductase screening be done? There we have different kinds of approaches. We have a sequence approach where the Roche people, for example, Dennis Wetzel, screened different kinds of imine reductases, in total 28, which accepted various aromatic and aliphatic as well as cyclic ketones to produce secondary and primary amines. And with their approach, they used the sequence. So they checked out, okay, which sequence is highly conserved and which not. They figured out that the N-terminal terminus is very highly conserved, and the C-terminus is not. So they chose this domain and did a clustering of the bacterial protein sequence. And with a special approach, they could identify the different kinds of novel IRED candidates. So they have different kinds of carbonyl compounds combined with the methylamine, which were quite nicely converted to the chiral amines with high conversion and enantiomeric excess. Also in a cooperation with Matthias Höhne, they could obtain the rasaglin. The R enantiomer of rasaglin is a very important product which is used in the pharmacy to treat the Parkinson disease. Another sequential-based approach was done by our group, therefore Verena, Stefan and Jörg. explored different kinds of activities of 20 new novel imine reductases. Therefore, they used the protein sequence homology of 4 confirmed IREDs, which are known from 2013. And since the first crystal structure was solved also during that time, they suggested to take protic amino acids here at this position, which are proposed to be crucial for the substrate protonation. And therefore they have this aspartic acid and the tyrosine here, and they searched the BLAST database with this criteria of the acidic residue as well as with the Rossmann-fold domain in this position. And they could classify different kinds of novel imine reductases as D-type branch for the aspartic acid residue and the Y-type branch for the tyrosine residues. Here they tested with these novel imine reductases different kinds of imine substrates. In the dots here they showed the conversion, so the higher the dots, the more the imines are reduced to the chiral amines. And also we have here an orange, which is the R enantiomer, and in blue we have the S enantiomer. And as you can see, these imine reductases have a quite nice conversion with all these investigated imines, as well as very good enantiomeric access. During this approach, they could observe activities at 1 milliunits per milligram, and they used as a carbonyl substrate 10 millimolar as well as with the amine. And they used the ADH to recycle the cofactor NADPH. And with this they used 5% of isopropanol for the alkyl dehydrogenase, which is a rather nice approach. Another methodology for the IRED mining was published in 2021 by Marshall and different other co-workers. And there they pursued a metagenomic approach where they sampled all different kinds of sequences across all the United Kingdom. Then they did a sequence analysis and selected different kinds of related protein sequences across all different kingdoms of the bacteria. Then they did cloning, enzyme expression, and with a robotic platform, they did a high throughput screening. Therefore, they developed an assay where they could detect NADP plus by a diaphorase mediated reduction of the tetrasomium salt, which led to the formation of this corresponding red-pink 4-Mazzan dye. And with this approach, they could identify 300 new imine reductases, which could also accept rather demanding ketones and amines for active pharmaceutical ingredients. Have a closer look in this photometric assay. They also used the target amine here to be as a substrate to observe it in a oxidative reaction manner. So here, as you can see, the amine product can be oxidized to the imine product, leading also to the carbonyl and the amine donor. And during this oxidative route, the red compound is produced, which can be shown here in these red hits here. And the numbers in these hits are from the reductive pathway. From the reductive amination pathway, they use the GC analysis, where they investigated in the conversion. So for example, sometimes you can also see that they have a positive hit on the oxidative route, but none from the reductive route, which is rather fascinating. So with this approach, they used different kinds of amines on the oxidative site to produce the imines, to check out which imine reductases are relevant. And there they figured out that they have best hits with this amine product leading to the imine substrate. So with this colorimetric assay and the GC analysis, they could have a very nice broad scope of different kinds of carbonyls and amines which were accepted by the imine reductases. And the best ones are listed here, where also scaffolds could be synthesized by these imine reductases with a rather nice conversion, also with a very good enantiomeric excess. So let's have a closer look towards the structural biology part. The imine reductases contain an N-terminus where the cofactor NADPH is bound. Then this N-terminus is connected via a helix, interdomain helix here, with the C-terminal domain, which is not so conserved like the N-terminus. The N-terminus contains a Rossmann fold, which have like alternating alpha and beta sheets, which constitutes the Rossmann folds for the NADPH binding. Here the first structure was solved in the bacteria Streptomyces canamyceticus. Then another approach from Aleko and Nick Turner was done here. They superposed two different kinds of crystal structures of the imine of one imine reductase. And here you can see in green, that's the crystal structure without the NADPH cofactor. As you can see, it's more on the outside this loop here. And when they did the crystallization with the cofactor together, they can find that this loop region here is further moved closer to the NADPH molecules. So it's like a lid which closes the active site, which is also smaller after NADPH is bound. Then they also co-crystallized this imine reductase with a racemic mixture, and they found out that the R-selective substrate is preferred, and probably also the tyrosine residue and the asparagine here, which are stabilizing the substrate, has a potential role in the active site. Another paper by Aleko later on was published where they crystallized the R-selective rasaglin, which is a pharmaceutical agent, with a reductive aminase, which is a subclass of the imine reductase. And there they figured out that the position 169, the aspartate, and the tyrosine in 177 are rather important for the active site, and they proposed that they protonate the substrate. And yeah, with this, they use different kinds of mutagenesis studies where they mutated the aspartate to alanine or the tyrosine as well, and they figured out that the activity of these variants were less prominent with the amine and carbonyl substrates compared to the wild type. Also published by Nick Turner and Gideon Grogan in the recent group, the first proposed reaction mechanism was based on this crystal structure here, where they co-crystallized the cyclohexanone and the allylamine in the active site, as well as the inactive cofactor NADPH here. So they proposed the tyrosine, aspartate, and asparagine as a catalytic triade. And in the first step of the reaction mechanism, the carbonyl and the amine is binding as well as the NADPH to the active site. And then the aspartate is deprotonating the amine, which is getting then more electrophile to attack the carbonyl carbon here. And this is stabilized by a tyrosine, leading to the hemiaminal intermediate, which is formed in this step. And afterwards, water is eliminated, and then the NADPH can deliver its hydride to do the reductive amination, which leads to its oxidized cofactor and our desired amine product. Imine reductases also have the potential to be used in enzymatic cascades. Therefore, Nick Turner group combined the imine reductase with an amine oxidase to do a deracemization of racemic amines. And here they enriched the S enantiomer with this cascade. Then another paper from France et al. 2016, they used 3 different kinds of enzymes together, and they used keto acid as a starting material, which is then carboxylated to the ketoaldehyde, which is then aminated by the omega transaminase, and spontaneous cyclic imine is formed in this case, which can be accepted by an R or S selective imine reductase, leading to these piperidines or pyrrolidines. And this was all set up on a one-pot reaction, which is quite fascinating. Since you also need to go with the flow with the enzymatic cascades, another research group published this paper here in Angewandte, where they used alcohols as a substrates, and they used the alcohol dehydrogenase to yield to different kinds of aldehydes, but they also put different kinds of channels to the imine reductase and transaminase. And the imine reductase and the transaminase here, they immobilized for the flow, and the imine reductase there, they implemented a glucose dehydrogenase for recycling the cofactor, of course, and they could obtain different kinds of primary amines with the transaminase, but could also employ a process to yield secondary amines with the imine reductase with a very good conversion with this approach. And with this approach, they could also further synthesize the methyl norbeladine, which is an intermediate for some alkaloids like galantamine which are used against dementia. Imine reductases can also be implied for industrial purposes. So here for the people from GSK had an approach to synthesize this demethylase 1 inhibitor from GSK, and there they used different kinds of racemic mixtures, and they could develop an enzymatic process. So they had like fewer steps, they had the kinetic resolution, and also improved green chemical approach compared to the chemical one, where they used a stoichiometric amount of sodium borhydride, which is not so great for the environment, as well as with a large number of solvents. So, what they did is they wanted to have kilogram quantities of their key intermediate of this GSK inhibitor, and they obtained it with a rather high yield, with 84% and 99% purity, as well as an excellent enantiomeric excess and an improved mass intensity. Therefore, they used the 3 different kinds of rounds of evolution, starting here with a Y type. They used different kinds of homology models, and here where they highlighted the amino acids which were mutated. So here in the beginning they mutated the tyrosine to serine and also changed some of the conditions. Then in the second round they ended up with 7 mutations for this M2 variant, and at the end they could obtain with 11 mutations the best variant so far, where they stated 38,000 fold improvement compared to the wild type, which is quite impressive for a kilogram scale. So to conclude everything with this fantastic imine reductases, they can catalyze the imine reduction and the reductive amination. Different kinds of amine and carbonyl combinations can be used, and these carbonyls and amines are rather cheap and easily available. Then compared to other enzymes, they have a rather high conversion and enantiomer selectivity, also leading to chiral amine products. And there are a lot of different kinds of papers reported that used sequence-based, metagenomic or structure-based IRED binding to detect new imine reductases. The reaction mechanism was shown in this presentation, where the group found out that tyrosine, aspartate, and asparagine is the catalytic triads so far, and they can be combined with different kinds of other enzymes, and also can be applied on a kilogram scale for industrial purposes. With this, I would like to come to an end, and I hope you liked the presentation.